so you can share your screen. And whenever you're ready, thank you.
Okay, great. Just to confirm that you can see my slides and hear me?
Yes, perfect.
Great. Okay, so hi everybody, I'm Orit. I'm an assistant professor based at CU Boulder. And just as a side note to the organizers, it's been an incredible experience. It's my first time in Alife, and there's just been so many good talks and activities. So it's really great to be able to contribute as well. I'm going to tell you today about our project called Data-Driven Modeling of Distributed Resource Sharing in Honeybee Swarms. I'm presenting it on the behalf of Golnar Garuni-Farad and Elizabeth Brady and myself. Okay, so, oops, this is, okay. So honeybees are a landmark example of eusocial organisms that have a very sophisticated way to divide tasks inside their colonies. For honeybees, a few examples include feeding the young, feeding the brood, constructing or cleaning honeycomb, or even going a little bit outside of the hive's structure to ventilate and guard. And all of these activities cost energy. The way the bees, some of these bees would actually get energy is through another caste of bees that are foraging, and they go and forage flowers, collecting nectar and pollen, and then bringing food in their social stomachs, and then disseminate it to the rest of the or several parts of the colony. This behavior is called Trophallaxis, and what's happening is that the fed bee that has food in her stomach is regurgitating some of this food and sharing it with other nestmates. And here is a movie showing that behavior. I'm not sure if my connection allows you to see that, but basically there is a bee on the right side that had access to food, and now it's basically feeding another bee that is more deprived and didn't have access to food. So of course, this process is very important for the survival of the colony, and it drew the attention of many researchers. Just to name a few examples, there's beautiful work from Ofer Feinemann's lab, who actually works with ants, but on a similar problem, and he can fluorescently label the food that the ants are consuming, and can see how it's moving between pairs of ants and in the network. Another recent example is coming from the group of Gene Robinson, who can actually track an entire colony of honeybees and follow their Trophallaxis interactions, and they can get some very nice statistics about when do these events occur. But from my point of view, what's missing currently is a mechanistic understanding of and a local set of behavioral rules that would reproduce some of these feeding networks that we see in nature. And as we are in the Alife conference, I also want to mention that there is quite a lot of interest in that kind of behavior in the swarm robotics community, where a known problem is how to charge all the robots simultaneously. So there's some interest in that kind of solutions in order to well, basically divide and conquer and have some of the robots charged and so on. The situation that I'm going to focus on today is the following. Let's say that we have a bee in our experimental space or simulation space, and it has a full stomach, and it just fed a deprived bee. Who should this bee feed next? Should it feed somebody that is very close by, or should it move around a little bit in space and then perhaps feed another bee that is further away from her? We can sort of think about this as a diffusive process, where what the material that is being diffused is the food, and the way the bees move around basically control that diffusive process. So in a very naive approach, we can just think about normal diffusion, where we know that the mean square displacement is a linear function of time, but with a certain coefficient, that is diffusion coefficient. So essentially the bees can manipulate that diffusion coefficient by moving more or less in between of these feeding events. There's no reason also to believe that it's a purely normal diffusion process. It can also be something a bit more sophisticated, like a super diffusion, where individual bees that feed other bees, basically these fed bees would start the diffusion process all over again in that specific spot. So we're kind of keeping this in the back of our minds while asking how local rules of movement and interactions will affect the global efficiency of food distribution. And the approach in my lab is using a combination of mathematical modeling and also behavioral experiments that we do actually with living honeybees. Okay, so we start with a model. It's a very simple and naive model where bees are just agents performing a random walk. It can be a pure random walk, it can be a correlated random walk, and we can actually control that by tweaking the probability distribution function of the turning angles in between steps of the random walk. Which is shown here as an illustration on the right side. So if that distribution is very narrow, then the bees tend to walk in more straight lines and they move further, so they have effectively a higher diffusion coefficient. So these are our agents, and then if bees encounter each other in space, we define that just basically by a threshold distance. Then they would exchange food, and for simplicity, we just define the exchange to be halving the food. So these two bees would actually leave this interaction with the same level of food in their social stomachs. So these are the social rules of behavior and movement. And then in every time step in the simulation, we repeat those. And we repeat them until we reach some convergence of the food distribution. We define this by looking at the amounts of food that each individual agent has, taking an average that and basically looking at the variance of that. So when the assumption is when the variance is below a certain threshold, then the food is more or less equally distributed, and that's when we stop the simulation. Okay, so here are some results that we can get from this kind of simulation. What you see here, this plot, the y-axis is the convergence time, how many time steps we needed in order to get the food to be equally distributed. And we show it as a function of the turning angle distribution width. So basically, when it's a very low value, then the bees tend to walk straight, they move further, they have effectively a higher diffusion coefficient. And we can see that similar to the intuition that I showed you in a few slides ago, that would result in a lower convergence time. In a sense, it's more optimal. So to show this in a slightly more visual way, here are a few examples of the agent-based model simulation. So on the left side is a situation with more mixing. On the right side is a situation with less mixing. The colors that you will see the agents colored by represent the amount of food that they have in their stomachs. And so here it's how it looks like. You can see in the case where there is more mixing, the bees move further away from their initial position, and we reach equilibrium in terms of the distribution of food much faster. Okay, so great. So we have this hypothesis, and we actually have the bees in the lab. So we go and check that with the bees. We worked with European honeybees, Apis mellifera L, and we had two groups of honeybees. Some of them were fed and they had access to food. And we marked them with a small pink dot. And then we also had a group of bees that did not have access to food, so they're the deprived bees. We set the ratio of these two groups such that there is a very small amount of fed bees, because that would basically stress the bees to respond in a way that would optimize the food distribution, and there's a higher chance that we will see something interesting in that scenario. So our experimental arena is two-dimensional or semi-two-dimensional, and the bees can just walk along this semi-two-dimensional arena. We're now looking at it from the top, and we record the movement and behavior of the bees for several minutes, actually up to an hour, and we can see how these full bees are moving, and we can actually track them with really simple computer vision analysis. So this is an example of such an analysis. So what you see here is the red lines are the trajectories of the fed bees, the full bees that had access to food. And it's overlaid on the average density that the rest of the bees were corresponding to. So we see that the fed bees actually move quite a lot, which is consistent with what we expected from the naive model. But actually, very quickly after that, they stop moving. So these lines, these red lines, actually look now more like dots. And to put this in a slightly more quantitative way, I'm showing here a plot where the y-axis is the average speed of the fed bees as a function of time. So they actually stop moving, which is completely counterintuitive if we think about the diffusivity argument. And so it's turned out that that's not the end of the story, and the bees actually do something else that is important for disseminating food efficiently. And it seems that they actually cluster. So here again is the same data, but I'm just showing the cluster analysis, where we basically clustered bees according to their density, and we can follow the formation of these clusters. And what is shown here in the plot, the y-axis is the cluster size as a function of time. The dotted line is the time point in which we introduce the fed bees. And we can see that before that, we get pretty small clusters of about between 10 and 20 bees. But then they significantly increase once we introduce the fed bees. So there is a coarsening and aggregation process that we wanted to basically go back to the model and include that and see if it would help us understand better what the bees are doing. So it did exactly that. So we use exactly the same model, but add one new rule where we add an interaction radius, we call it R, neighborhood, where if bees are separated by a distance that is smaller than that radius, they basically turn their movement direction, their heading, and they start moving closer to each other. That's it. That's the only rule that we added. And the rest of the rules are the same. I'm not going to repeat them due to time, but we basically do exactly the same process where we follow these behavioral rules, let the simulation run for many time steps until it converges. So this is what we see here in this modified version, we call it the data-driven model. The y-axis is again the convergence time like before, but I'm showing it actually as a function of the attraction range. And we can see that at very small values of the attraction range, we get this plateau value where nothing is really happening, but then at slightly larger values of the attraction range, we get this sweet spot that gives us optimal convergence time. And I'm actually showing here 3 plots that show different values of the turning angle distribution. And this is just to illustrate that actually I think we get a much higher effect, stronger effect with the attraction range in comparison to tweaking that movement of bees that I talked about before. Okay, and again, just to visually illustrate it, here are example of these 2 simulations, one with attraction on the right side and one without attraction. So we can see visually that we get these clusters, which is encouraging, but we can actually even more systematically compare that to the experimental data. So now if we plot the cluster size as a function of time, what you see here, the red plot is the experiments. The blue plot is a simulation with attraction set to the attraction range that basically get these 2 plots to fit to each other the best. So this is the best fit. And then the green one is a simulation without any attraction. So of course we don't see any clusters. The cluster size is close to 0. But it's pretty interesting that we converge on an optimal value that gets the experiments and the simulations fit to each other best. That value is about 2 centimeters. And that actually not that far away from the optimal value that we see in the convergence time versus attraction range plot. Okay, and then just as an outlook, I want to mention that we're also interested in how robust that mechanism is, and one way to manipulate something about the conditions of the experiment that the bees are experiencing is changing their density. So basically have more or have less bees in the arena. Changing their density could significantly change the probability to encounter each other. So we are repeating this kind of exercise for different densities. This is still work in progress, but what I can tell you as preliminary results is that it becomes more important to tune the attraction radius to the optimal value at low densities, which makes sense because that's exactly the situation where the bees are less likely to encounter each other unless they are attracted to each other using these local interactions. Okay, so to summarize, I showed you that we built a Trophallaxis inspired agent-based model, which is designed and also somewhat validated using lab experiments with honeybees. Our main conclusion is that a more tightly constrained random walk with short range attraction can increase the efficiency and provided a local set of rules that would reproduce that behavior. There's more information in the paper proceeding. It's also on the bioarchive. And I just want to acknowledge everybody in my lab, especially Golnar, who is a graduate student who led that work, and Elizabeth Bradley, who provided us immense support and feedback. And thank you for your attention. I'm happy to take questions if there's time.
Thank you. That was great. We're a little over time. I didn't want to stop you because this was so interesting. So there's one question in the chat, if you could answer very quickly. Early on in the talk, Ian was curious about if you could elaborate on the quantitative properties of super diffusion. Is there a probabilities distribution for the process that you can solve?
You think you can find equations for these anomalous diffusions? That's my really question.
Yeah, so I mean, I just pose it as an illustration for another kind of process that could happen. And we haven't tried to systematically compare what we're seeing to super diffusion. But I think the rationale is there. So we might be able to do that, but we don't have any specific equations.
And sorry, I know we're over time. I just wondered if you could just quickly recap what are the summary statistics of your experiment? I saw convergence time, you talked about diffusivity. Is there anything else that you're sort of summarizing the data in terms of?
I would say another important thing is the cluster size. So we can see that the bees create these big clusters early in the experiments and also early in the simulations, and that seems to be really important for efficiently distributing the food.
Thank you. And I'd invite you to look in the chat. There are a lot of other questions I think people were interested, and we can keep the conversation going on.